I can show it to you.
Yeah.
Hey, Cas.
Uh, Hey, how are you?
Good. Good. It's like we redo the eyes.
Yeah, we're gonna the last 15 minutes. We'll
Mm,
revisit that. No.
yeah. So, how are you doing?
I'm good. I'm good. Thanks for
Mm,
inviting me.
I mean, thanks for coming. I'm really, really excited about this podcast.
Yeah, me too.
Yeah. So, would you like to introduce yourself?
Sure. Yeah. So my name is Cas Wognum. I'm actually not a PhD graduate. So I'm not a doctor. You can just call me Mister, I guess. I'm a computer scientist by training from the Netherlands, and
Hmm.
I moved to Montreal about 4 years ago, and I've been working in machine learning for drug discovery ever since.
Hmm. What got you into drug discovery?
It's an interesting question. I fell in love with computer science when I realized I could create systems that are better at something than I am. And I can remember this specific class where we had to build an AI to play checkers.
Hmm.
And I was debugging it, and I don't know, I kept tweaking it, and it wasn't working. It would sacrifice all of its pieces. And at some point I was like, well, I'm just gonna finish playing. And I'm not a good checker player at all, right? And so what
Hmm.
ended up happening is like it kept sacrificing its pieces, and I didn't understand why. And at some point, like
Hmm.
6, 7 moves ahead, it would just like everything would line up perfectly where it could take all my pieces back and then some in one turn. And it would kick my ass. And so it's that class, that project that really got me excited about AI, because it was like, okay, well, or computer science in general, like I can build something that's better at something than I am. And with that excitement also came a sense of, I guess, a sense of responsibility. Like that's a heavy
Hmm.
word, but like a sense of responsibility where I was like, well, if I can do that, I want to use it to do some good. And that for me could have been a wide variety of things. I've been very excited about filmmaking for a long time, and I've explored going down that path. That didn't really work out, but that could have been it. Like entertainment I could have felt like I've made a difference by making nice movies, or like that would have been enough for me. But then it like life took a different turn, and I stumbled across drug discovery. And really, the reason is I wanted to come to Montreal because my girlfriend
Hmm.
is from Montreal, and I'm from the Netherlands, so there was this long
Yeah.
distance thing. That's a whole other story. I'm not going to get into that. But like, I had a personal motive to come to Montreal, and so I visited Mila, which is the main
Hmm.
like eco center for machine learning research. And I can just remember taking a picture of all the logos on the wall. And just at home, I would just go through them one by one. And I came across Valence Discovery at the time, and I was like, that looks like a nice company. Let me just see. I've had 1 year of biology. I had 1 year of chemistry. That's all. But I was like, I'm just curious. Like, and I wrote to them, and sort of it went on from there.
Hey everyone, this is not an ad. Go to blueword.com/in, that's BLUWR.com/in, like inside, and write about something that makes you think.
Oh, sorry. Cold email.
Yeah, I wrote a message to well, one of my colleagues on
Yeah.
LinkedIn. I just wrote a message
Yeah.
like, hey, like your website looks great. It sounds really exciting
Hmm.
what you're doing.
Hmm.
I don't have a background, but I have a pretty good like education. Like, is that something that could be of interest to you guys? And they were small enough at the time that they were like, oh yeah, like come on by, we'll chat. And then
Nice.
from there, one thing led to another. Yeah.
Nice. Was it which person? Was it?
It was Dominique,
It was
Dominique
Dominic. Okay.
Beaini. Yeah,
Yeah.
yeah,
Mm.
yeah.
Yeah. Dom is a great guy.
Oh yeah, no, he's
Hmm.
awesome. So no, I think he took a chance on me.
Hmm.
Again, like it was a smaller company, it was a different time,
Yeah.
but yeah, he took a chance on me, and then I'm kind of happy he did, because now I'm here. We're 4 years further, and
Hmm.
it's such an exciting space. It's such a nice time to be a part of it as well.
Yeah. And that was before Valence got acquired by Recursion.
Right? Yeah,
Yeah.
yeah. So it's like also in terms of like the like the path that the company has been on, and that we have as an organization have been on. It's just a really interesting. There's a lot that
Yeah.
happened. I mean, like Valence itself grew a lot in
Hmm.
the time that I was there.
Yeah.
And then like being acquired by Recursion, and Recursion is just an ever-changing environment. Like that's an even like
Hmm.
it's been a very interesting time.
Yeah. Yeah. I think we should do a podcast one day and just about the story of Valence.
I'm sure that like Dan or Terrence, yeah, they're very, very open about sharing. It's a super
Hmm.
interesting story, I think.
Yeah, it definitely is. I remember that when they had like this very small office at Nutman House.
Exactly. Yeah, yeah. Oh, at Notman House even. Yeah,
Yeah,
yeah. So
Nutman House. Yeah.
I joined when they just moved into Mila, but it was also this very small office,
Hmm.
and it's like it's tucked away in a corner somewhere. It's dark, like it's not very nice to go there. So yeah, like we've moved out. They've moved twice in the same building now, and like it's just yeah, it's a nice. Again, I'm repeating myself. It's nice
Hmm.
to have been on that trajectory. Like you see a lot in just a small amount of time.
It's a nice arc. Yeah.
Yeah,
So you got so basically I'm simplifying, but you're a computer scientist, discovered AI, and then you're like, it would be nice to save lives.
yeah. And you're simplifying it, but I think there's a
Mm.
certain naivety that a lot of computer scientists, including myself, have,
Mhm.
which is like, oh, I can throw machine learning at this and I'll solve drug discovery, right? Like
Mhm.
it's almost like a bit of a running gag with some of my colleagues. It's this idea of like, okay, we're at this point now and we're going to solve drug discovery next month. This idea of like
Yes.
solving drug discovery, it's just
Mhm.
like I don't know, it's ridiculous now that I understand what it actually sort of implies. But like when I entered the field, there was this naivety, and it's I mean it it can be a little detrimental to the discussion that the field is having, right? But it's
Yes.
also nice to have people come in with that energy
Mhm.
of like,
Mhm.
let's just solve it. We can throw some computers at it. We can compute this, like
Yeah.
that are not burdened by like everything that has been tried, and they just come in
Mhm.
with this fresh mindset, and they're like, let's just try it. Like, why not? And so that was me. And
Definitely.
like you summarized it, but
Mhm.
actually, I do think that I sort of had that.
Mhm.
I had that mindset of like, let me save some lives. How hard can it be?
Yeah. I mean, we can make fun of it because we did the same thing. So
Y-
there was also my mindset.
yeah, yeah.
Yeah.
And again, I
Mm.
don't think it's all bad, right? I'm even to this day, like
Mhm.
I love the excitement. I love it when there's like we have an intern or there's someone new that like just graduated. Like I love the passion and the conviction and the let's just do this
Mhm.
mindset.
Yes.
But there is a reality as well, which all of us are at some point confronted by. Is like if we're a little
Mhm.
bit down, like a little further in our adventure, we realize okay, there's a lot of moving pieces here. Maybe like simplify,
Yeah.
like scope it down. But anyways, like
Mhm.
it's a good balance between those two. You don't want to be you don't want to be too pessimistic either.
Yeah, yeah, yeah. And I think you're absolutely right. I think that's what we computer scientists bring to the table.
Yeah,
Unbounded optimism.
yeah, yeah,
Yeah, but because that was basically my story. When I finished my master's degree, I was at Lisa, like the ancestor of Mila,
yeah,
and I didn't want to work on image recognition. Didn't interest me that much, and I really didn't want to work on ads, targeted ads, which was like the
yeah.
big thing back then. I mean, it's like almost 3 years ago now. And then it was like, yeah, let's go and save lives.
Yeah,
Yeah.
yeah,
Exactly, how hard can it be? But it's actually very hard. But the fact that you bring that enthusiasm and that energy actually makes it possible in the end. Yeah,
I think so. Yeah,
yeah.
and like even if you don't move the needle
Mhm.
like as much as you maybe had hoped initially, right? Like
Mhm.
just coming in, setting these audacious goals, and like just shooting for them. I do think like I don't know, like if you set like a very safe goal and you sort of set it here, right, and you make that
Mhm.
goal, that's where you're gonna be. But if you
Yes.
set the goal to be like all the way over here, and maybe you fall short and you're only here, you're still a long way up from if you would have just set like a safe goal, right? It requires you to not just think in incremental solutions, but to really
Mhm.
think about like what's the leap forward that we can take? How do we rethink things?
Yeah.
Anyways, it's a balancing act. You don't want to have too much like naive optimism. Like at some
Mhm.
point, that's not helpful anymore either. But you also don't want to have too much realism. That's like so it can become
Yeah,
pessimistic.
exactly.
Yeah,
Sometimes what we feel is realism is actually not realistic at all.
exactly.
Yeah,
Yeah,
it's
yeah,
being overly pessimistic um, is not being a realist.
exactly. Yeah,
hmm
yeah,
Yeah.
yeah.
So, what was the first AI algorithm you developed?
Oh, so it was like that was the checkers thing.
Okay.
And so it was this really simple Monte Carlo tree search.
hmm
There was no deep learning involved whatsoever. So it was really just an efficient search algorithm.
hmm
Um, but yeah, I do think it's one of the coolest things to date. Like it,
hmm
it's cool because it's a product. It's something you can
Yes.
interact with, right? Like I can play something I've built and it can beat me.
hmm
And I always thought that was really cool.
Yeah.
Yeah, so
Yeah.
that was really the first AI, my first like encounter with AI or
Yeah.
machine learning at the time. It wasn't even called machine learning, I think. And
Um, I mean, it was
uh,
that's yeah. So because what we call AI today is actually a subset of AI, like the deep learning algorithms. But the
yeah,
AI used to be a much broader set of of tools.
yeah.
Yeah.
I did my master's degree at the University of Utrecht in the
hmm
Netherlands, and they had this very multidisciplinary um, view of AI. And so we
hmm
would indeed not only learn deep learning and machine learning, but we would
Yeah.
also learn um, like like the more classical reasoning algorithms
hmm
over using ontologies or whatnot. A lot of these search algorithms.
hmm
We even have classes on like ethics and like the
Yeah.
ethics of AI. And so it was very multidisciplinary. And I think when I followed the study at the time, I at times felt like I was at a disadvantage because of it, because I would see some of my colleagues, my students that I would meet from the University of Amsterdam or any North American university, really, they were really focused on machine learning and deep learning,
hmm
and so I was like,
hmm
I feel like that's where all the excitement is now, and I'm not really, I don't know, I'm
hmm
not really like learning as much about it as I wish. But I think now that I'm a couple years into my career, I'm quite happy that they structured the program that way, because you're never gonna learn that on your own. Like right now,
hmm
for me, it's quite easy to learn about deep learning because it's what I'm using in my day-to-day life. But
hmm
I would never. It's really hard for me to build up the discipline to learn about something that's sort of like adjacent to what I do.
hmm
But I don't know. Whereas I do find that in my daily job, like the context that I gathered and the historical perspective that I gathered from having that multidisciplinary study is helping me tremendously
hmm
in just my like structuring my thinking.
Yes.
Um,
Yes.
so yeah, at the time it felt like I'm falling behind. Whereas now I'm like, no, actually, like catching up on the machine learning piece feels doable. Like, of course, there's so much going on. You can't be an expert in everything, but like it feels doable. Whereas
Mhm.
it's really hard to learn about everything else that's not as popular right now.
Yeah, yeah, definitely, definitely. And I kind of feel the same thing. I mean, I'm somewhat of an older generation when it comes to because I came basically I started my AI journey around 2007, 2008. So I saw the previous the previous iterations, and I feel extremely fortunate that I was able that I got that training,
Yeah,
especially like hardcore computer science, which I think is lacking at the moment. Yeah,
yeah, yeah,
yeah,
yeah. Um, yeah, I agree. It's just like I did um like in terms of hardcore computer science, I did
hmm
classes on like optimizing like algorithms and data
yeah,
structures and using hardware and hardware acceleration, and
hmm
like honestly it's not top of mind anymore. I wouldn't be able to
yeah,
like do the exercises I did in that class, but just knowing roughly what's happening behind the scenes when
hmm
you're training your model, and you get to the point where like, okay, now I need to train across multiple GPUs, or I'm becoming memory bound, or I'm becoming compute bound. Just
yeah,
having a concept of what that means, and what, like, having a
hmm
rough idea of the tools in your toolbox you can use to
hmm
circumvent that is incredibly useful.
yes.
Yeah,
It's incredibly useful, and when you compare that to some of the newest generations, and some people who do AI today, they're like the youngest people. They have like they started their journey and there's hugging face.
Which is great.
Which is
It's like,
great.
yeah,
Yeah.
it's like, it reminds me of like the evolution of like programming languages, right? Like
Hmm.
it's a new layer of abstraction, which makes the field accessible to a broader audience. And that's great. We need more people that can do machine learning. Like that's awesome. I welcome everyone, right?
Yes.
But that doesn't mean that the skills that are needed to build a hugging face,
Hmm.
like those skills are still needed too, especially in
Yeah.
certain scenarios.
Exactly. That's also my experience is that there's this somewhat yeah sometimes you have this illusion that you know how to do the thing but you actually don't, like using Python like high level language, but you're not I mean um, unless you know how you have an idea of how to build Python itself. That would be like a starting point. Like, could you make something like Python, and let alone something like PyTorch, let alone something like Hugging Face,
Yeah.
let alone, and if you go like lower and lower and lower, like something that actually interfaces with the machine, and in an optimal way, because now we also have like we have big machines, we have a lot of resources, but it doesn't mean that it's free. It's not because there's a lot of it that it's free, and having that understanding, just not even understanding, just like an idea of what's happening behind the scenes is actually extremely beneficial, especially when you get into a production environment.
Yeah, and not even production. I mean, yes, production environment, but I've also found myself, when you're doing frontier research,
Mhm.
um, by design, you will often find yourself in a situation where the framework can't yet do what you want it to do,
Mhm.
right? Because you're doing something entirely new.
Yes.
And so at that point, you start running into the limits of the framework, and it can be really confronting if you then realize, like, I actually don't know what's happening under the hood. And it's not just like, I know we started this by saying like I'm happy I've had that background, but I mean, for me it's
Mhm.
the same. Like I use a lot of these libraries, and I don't know to the full extent what's happening underneath the hood because I don't need to. And that's all right. Like I think that's perfectly fine. It's just when you run into those moments where you're like, okay, now then I'm really happy that I have the context
Yeah.
to understand such a library a lot more
Mm.
quickly than sort of really having to start from scratch. I have that foundation to build on.
Yeah, exactly. That's a good introduction for Polaris.
Yeah. Well, actually, I think it is a nice leeway. Like, I think a lot of the stuff we talked about, it feeds into, like, it's
Mhm.
my journey, but I think it's a journey for a lot of people. Yeah,
Mhm.
like we were saying, right? Like this sort of unbounded optimism.
Mhm.
Um, and so, yeah, well, when I joined Valence Discovery at the time, my first project was on out of distribution generalization.
Mhm.
And I come into that with a certain naivety of like, okay, we're just going to split the data set. You'll tell me how to do the split. And then once we've done the split, I'm going to do the modeling. And then it turns out that actually, even how to do the split is not, there's no, the answer is it depends. There's no
Mhm.
perfect answer here. And so that led me down this path where I started questioning everything in terms of evaluation. And I just realized that, like, time and time again, I would talk to these incredibly generous colleagues of mine that would share all their knowledge with me, but and they're at the top of their field. But even for them, the answer very often was it depends. Like we don't have like clear cut guidelines for you right now. And so I was really lucky to be part of a company where there were all these, like, I was part of a group of people that had interdisciplinary expertise. Um, but so biology, chemistry, computer science, physics, like there were a lot of people that had expertise on multiple of those domains, or at least one of those domains, and I could talk to all of them. But it was also an industrial setting, and there was an industrial experience. There was an understanding of how these models that we're developing are actually used in the real world to drive impact. And so I was incredibly lucky to be in such an environment that I could actually formulate benchmarks that we believe to be impactful. But what I realized is, like, if I wouldn't have had that environment, which is the case for many people, right? Like
Mhm.
many people entering the field many academics, they don't have access to either the interdisciplinary expertise or just the industrial experience that you need to formulate impactful benchmarks. And I want to be very clear, like that's no one's fault, right? It's not like people don't care about benchmarking. Like, no, I think everyone has good intentions. Everyone wants their work to be impactful, but it's just an incredibly hard thing to do. And there's not a lot of people in the world that can do it. And you really need to be part of an environment where you can find all these little pieces of expertise that you need. Please
Yeah.
go ahead.
Yeah, definitely. And what I think also is from my experience is it's not just getting people with it's not about getting people with different
whole
expertise in
globe
the same room.
wou
Actually,
mentar
the
algo
expertise has to be
que
inside the
tu
people. You need people with
consultou com
different sets of expertise.
Yeah.
That's the only way to allow for efficient communication.
Yeah, it's yeah, I think communication is key. Like you're right. Like you don't just want to you don't just want to bring people together and that fixes everything.
Mhm.
It's this you need this, which currently is still a rather rare expertise that not a lot of people have, but you need this fluency with different languages of different scientific disciplines.
Mhm.
And yeah, so you're right. It's not just about bringing people together. You need to learn to speak each other's language.
Yeah.
And so what we were seeing is like there was just a lot of benchmarks out there that for one reason or another, and again, not because of anyone's like bad intentions, but that for one reason or another just didn't really like measure what we were actually interested in measuring. And this was leading to a gap between perceived progress and real world impacts. And that's the problem that we set out to solve. And that's that computer science optimism again, but like
Mhm.
that felt solvable to me. I'm like, okay, we can do that. Turned out to be a lot harder, but yeah, that's sort of how the project started.
Okay. Yeah. And I think it's an amazing project because just So to give a little bit of context, for example, we have we ran some benchmarks in my lab, and some of the models that have been published for drug discovery are actually not as good as standard models that you could have on scikit-learn.
Yeah.
Yeah.
I used to joke about this with a lot of my industrial peers that like ECFP fingerprints with random forests are still state of the art baselines or state of the art models on like a lot of the benchmarks out there.
Exactly.
And the reason we don't see them in papers is because people don't want to compare to them because they know they'll lose. That's maybe a bit of a skeptic take, but it is. I think there is a reality to it where like like a lot of like if you just train a scikit-learn model with basic settings and you pick a basic representation for a molecule,
Mhm.
you'll typically get really far.
Yeah, definitely. You can really do that. Same thing for single cell. There's a paper, like I think a Systema paper just came in, and a lot of the models are you can beat a lot of the models with just averaging the gene expression.
Yeah. Yeah, exactly. Like the mean baseline in like single cell research, it's just incredibly hard to beat. And again, I like to believe, and I do believe that most people have good intentions, right? And so I don't think people are out there scrapping results. Well, there might be some, but I like to believe that the majority of people they don't intentionally scrap results because the results
Yeah.
don't look good, right? Like I think most people want to drive impactful research.
Yeah.
But it's about knowing, like if you never worked in single cell or you've never worked in property prediction, chemical property prediction, like, and you come into the space with your computer science background, like, is that the baseline you're going to be thinking of?
Mhm.
Probably not, right? Like, you need a little bit of domain expertise to know that a mean baseline is good because it's not trivial. It's not self-explanatory that model will do really well.
Mhm.
Yeah, it's just about like setting, like, helping people have like calibrate people their expectations. And I think if it becomes widespread knowledge that like the scikit-learn baselines do really well, on molecular property prediction, which I think it is by now, you'll see more people use them in their papers. And the more people read those papers, again, that awareness will spread, right? But if I
Yes.
read a paper and I see only like deep learning methods and I want to continue down that research, like that's the methods I'm going to compare against, because that's my frame of reference.
Yeah.
Yeah.
Yeah, exactly. It's not because it's not that people are ill-intentioned. It's just an artifact of having a lot of people from computer science jumping into the problem, but maybe they did a little bit more domain knowledge.
Yeah.
That's and also there's so many papers that it's not like it wouldn't be obvious to me. For example, if I was just starting in drug discovery, and I build a model and I want to benchmark with my model, scikit-learn wouldn't be my first choice. I wouldn't think of that.
No, but
Yeah.
that's like, if you're trained in deep learning, that's also not really what you're trained to do, right? Like if you look at computer vision, I'm taught time and time over again throughout my studies, I was taught that the ImageNet moment, at that point, all of a sudden we realized that deep learning is the new thing. And like all those old baselines we used to do, they don't really work. That was sort of I'm of course simplifying, but like, yeah, like that's the message I would get a lot. So like when I'm then faced with a new problem, of course, that's where my mind goes. It's like,
Yes.
oh, right, okay, well, I can skip all of scikit-learn because in ImageNet it didn't work, so it probably isn't going to work here either. The reality is actually that we're in a very different space,
Yeah.
and we need to
Yes.
be aware of what differentiates our space, drug discovery, from like image processing or natural language processing. And I think once you start realizing the difference, then it's like, okay, wait a minute, maybe those scikit-learn baselines, actually they do make sense, because we have much smaller data sets, for example. So yeah.
Yes. Yeah, much more data sets and data sets that are of different nature also. It's not because something works really well on text that it will work as well on proteins or molecules.
Exactly. Like it's about understanding your data and then figuring out, what inductive priors fit that data. And I think again, like that's easier said than done, because understanding the data in a domain like chemistry or biology or physics, people spend their entire academic careers to
Yes.
understand data. And so if you're coming into the field without that background, without any background in biology or chemistry or very little, you can't be expected to know all that, right? And so it's like when I train a computer vision model, I can look at the outputs and it's very intuitive. And I can say the model does well on pictures of dogs, but it doesn't do well when the dog is very close to the camera, but it does do well. Like I can, this is a really easy task for me to understand. If I do the same in chemistry, like I'm at a loss. Like I look at a molecule and it will tell me a solubility prediction. I don't know if that's true or not. It's like I don't even know where to look at, right? Like, I mean, I've built up some intuition after working in the space for 4 years, but when I joined, like, it was just like, I don't know, like I would just predict a bunch of things and then show it to my colleague or compute a metric that I was told to compute, but I don't have the knowledge, and I think I can't be expected to go through and like inspect the quality of the model. Yeah. Who? I think I lost you for a sec.
Yeah, I lost you too for a second. That's okay.
Okay. All right. Anyways.
Can just keep going.
Yeah. Right. So I think that was the observation, right? The observation is formulating a benchmark requires interdisciplinary expertise, and it also requires industrial experience. It understanding like how these models are used and deployed in practice. And there's very few people that have that, and there's no easy to follow best practices. And so what our goal is like, let's bring together a group of people from across the industry. So we have representatives from various different pharmaceutical or biotech companies. Let's bring all of them together, and let's work on papers with guidelines, so it's best practices on how to do benchmarking in drug discovery, specifically machine learning for drug discovery. That was sort of the idea. And the idea was, once we had those guidelines really specify the what, like what should you be doing in benchmarking. But we didn't want to stop there. We didn't want to stop with like publishing a paper and then sort of like calling it a day. We were like, once we have those, the what, we also need to make it incredibly easy to implement those best practices in your day-to-day research. So that's what we like. That's the how, and that takes on a variety of forms. I think the most like flashy of which is the Polaris platform, which we can talk about more at a later point. But it's important that we don't stop at writing a paper and we sort of like we like that's it, right? Like we publish a paper and then we expect everyone to adopt what we are suggesting. Like no, we need to go. We're asking people to change their behavior. It's not always self-explanatory why that's needed, and so we really need to make it as easy as possible for people to do so. And then there's one final piece, which is we also have organized several community initiatives. And the idea there really is it's sort of the why. Like we want to create incentives for people to start adopting these guidelines. So we want to make clear what to do. We want to make it as easy as possible to do so. And we want to create incentives to do so. And so creating incentives, that's a bit of it's a broad category. There's all sorts of things. Like I think us talking right now is a way of like creating incentives, just sharing awareness, right? But I think the important part is that we just are aware that we need to have a follow-up strategy. We can't just publish our papers and then take our hands off it and assume the community to run with it. There's like years of expectations and standards that have been established that need to be disrupted, and that takes more effort.
Yeah, definitely. We can move to Polaris if you want to. Yeah. You start.
Yeah. So I think so. The idea was right. Like we bring together these steering committees. Initially, we were like very ambitious. Again, the computer science optimism, but like we were like, we're going to do this for all of drug discovery. Well, that proved way too big. So we narrowed the scope all the way to small molecule property prediction. And we have a really nice group that we brought together, representatives from J&J, from Nimbus, from X Relay, Pfizer, AstraZeneca, like all the big names, or almost all of them. And we've published 2 papers together. And so the first paper was in Nature Machine Intelligence. It was a correspondence. It was really just a call to action. We were just saying like, hey, we think something needs to change in how we do benchmarking. This is roughly how we think we can approach it, and we need people to help us out. And then the second paper was a much more technical paper, which was on like our first effort at putting such a paper forward on like how do you compare your methods in a way that reflects the reality of drug discovery. And specifically, again, for small molecule property prediction. So that's the scientific collaboration here. We also have a sort of a product side of things. So we have the Polaris platform. In its current state, you can think of it as a sort of a Hugging Face. I don't want to say clone, but I think it's the best description. Like it's a Hugging, like it's in a similar vein as Hugging Face or Kaggle. However, the reason we decided to build it from scratch rather than relying on existing services is that we wanted to have the control to implement those best practices directly into the platform, so that whatever we come up with, someone can come to the platform and use those best practices in their day-to-day research with a couple clicks on a button. And that was really the main driver here, to like make those guidelines as easy as possible. So yeah, I think those are sort of the 2 main pillars. I mean, we can talk about both, but I think the paper is probably a good starting point.
Yeah, we can start with the paper. And also, what got me interested in Polaris, I mean, that was the one, the summer school. That's when I discovered Polaris. I think it was last year, right? Is it? Or
last summer.
Or last summer, yeah. It's the fact that it's, to my knowledge, I think it is the first platform that's so well done for drug discovery, where you have the data sets, you have the metrics, you have standard benchmarks.
Yeah.
Yeah.
Yeah, yeah. I do think there are for other domains there are a lot of platforms that look like it. Hugging Face is a big example, but you have smaller ones. Like in single cell, you have the Open Problems platform,
Yes.
which you might have seen, which does something similar. We have relations with a lot of these other platforms, and I think the work that they're doing is like incredible. Like I think we need more of this, and I think these solutions can coexist. Like I think they target different audiences. Like if I compare to Open Problems, for example, Open Problems seems to target more the biologist, whereas we are more targeting the the computer scientists. And like right, like you make slightly different decisions, but both can coexist. So yeah,
Yes. Yeah, both can coexist definitely, and we need something like Polaris. I think it's I think as a computer scientist, let's say it this way: as a computer scientist, having a platform like Polaris makes my work much easier and much more streamlined.
yeah. I think
Yeah.
you can
Mhm.
like. Well, do you want to talk about the platform first? Maybe
Yeah.
now that
Let's,
we're on the stream. So
yeah.
I think
Mhm.
one of the things that's nice is like if you look at like the main API example on the website, right? It's really it's computer science. Like we're targeting computer scientists. We want that like in 2 lines of code, you should be able to get a high quality data set. You should be able to get the benchmark associated with it with a good split. You should be able to like you shouldn't have to worry about data curation or like filtering out data points or missing values. Like it should just sort of work, right? So yeah.
Yeah. This is it. Can you see it?
Yeah. Yeah.
Yeah. It looks good. It looks really good.
Yeah. So like if you click benchmarks, for example, in the menu at the top, and then you click on try it out on the left, the yellow button, like this is the main code example I'm talking about, right? And so for us, this is really aimed at computer scientists to the point where we'll have biologists using the platform who are sort of or chemists who are sort of frustrating, or like chem informatics, right? Because they're like, can I just get a CSV? Like I don't I just want to have a CSV. I'll do all the pre-processing myself. And I get it. I get it. Like if you have the expertise, like we're simplifying things, but if you have the expertise, you should, of course, you want to have full control. And so yeah, for us, it's like the goal is not to provide people full control. The goal is to make it really easy for people to do the right thing. And so we're much more prescriptive. But that does lead us to create an API like this, where it's like just a couple lines of code, and you go through the entire benchmarking API.
Yeah. And so what happens when you do submit, when you upload to Hub?
Yeah, so all the results are stored on the platform as well. So you can, for example, if you close this pop up and you click, for example, on the very first benchmark, so the runs and posts benchmark. Yes.
So
The top left.
This one. This one?
No, the top left.
This one? Runs and poses? Yeah.
Yeah. So if you scroll down a little, you see here's the leaderboard, for example. So if you upload your result, you can directly upload your result to the platform, and it's stored there. And there's a lot I want to change about this, especially once we get into the paper. I think just this leaderboard, the way we're showing it right now, like I really want us to move past that. But the nice thing is that at least these results are all comparable, because we hard code the split, like the literal indices that make up the split. We hard code the data set, like it's immutable after being uploaded. And so everyone that uploads on this benchmark, those results are comparable to one another. So I don't have to go and rerun all these models on the benchmark, because I can trust, as long as I use the Polaris API, that my results are comparable to what I'm seeing on the leaderboard here. Yeah. But so I think it's worth saying, like this is the platform. It's the most flashy part. It's interesting for people it's a polished product. It's a software product. People can click around on it. People can use it. It simplifies their lives. Like, it's nice to see some adoption in the community. We're starting to get some citations. But like, this is the flashy part. But I really think of Polaris as a scientific effort first. And so it's maybe an interesting like bridge to get back to the paper, which is the platform. If we were to just build a Hugging Face clone, we're not achieving our goal. We don't want to do more of the same type of benchmarking. We want to really enable a different type of benchmarking. And so that's why I think of Polaris as a scientific project first. It's we need to figure out what those benchmarking guidelines are. And I don't have that expertise to do that by myself. No one does. No single individual knows all the intricate details of how to do benchmarking. And that's why we bring these groups together to come up with to write these papers. Yeah. Do you want to switch to the paper?
Sure, but I'd like to talk about the data sets first.
Oh, sure.
Yeah, so because this is, and the benchmark aspect is super interesting, but there's also the fact that you've created a space where researchers can easily access well-conditioned data sets. And I think this is also like super important.
Yeah. So it feeds into the benchmarking piece that we described earlier, which is if we want to have a benchmark and we want to have comparable results, we need to have control over the data set. We need to make sure that people aren't pre-processing their data set slightly differently, because of which we can't compare one-to-one anymore. There's like we were saying before, like data sets in drug discovery, they are of a different nature. They're much more heterogeneous. They're smaller. They tend to be imbalanced. So even removing just like 3 or 4 data points, like a handful of data points, could have a drastic effect on performance. And so we needed to be in control of the data set. So that's one thing. We needed to have a data set artifact of some sort. The other piece here, and that's like one of the reasons, like one of the early hints as to why we're building this from scratch, is that little certified toggle just next to the search bar. So we want to make it really easy for people to find high quality resources. And so we have a review process which lives on GitHub, where we ask different experts to chime in on the quality of a certain data set. Now, by default, that toggle is enabled. So if you're just a computer scientist, you want to get some hands-on experience, if you come here, you'll see only data sets that we have already reviewed and that we believe are of a high quality. Or not just us have reviewed, we also invite others in the space to chime in. And it's on GitHub, so if anyone else wants to chime in, they can. So yeah, that's part of the data set piece. It's like, one, we needed to be in control, but the other piece here is it's a first proof point of like, this is why we're building it from scratch. We want to be able to have filters like these and to incentivize best practices by default. Make it really, really easy to do the right thing and really hard to do the wrong thing.
Mhm. Yes, and also the reason why I really like the data set aspect is I think it even goes beyond the benchmarks. It's if you're working, for example, on ADMET, or for example, we have antiviral ligands. If you're working on EGFR, you can download a data set, for example, that has been certified, and so you know that you're working on something that's good quality and that you can base your research on. And yeah, this is not obvious for someone coming from computer science, for example, if you want, because you can just go and download data set, but you as you said, it takes usually a lot of expertise, and expertise also from different people to be able to assess the quality of the data set.
Yeah.
So in that case, you have clean data sets.
Yeah, exactly. And I mean, the review process in its current state is very much a proof of concept, right? It just lives on GitHub. It's free flowing text. Like, there's not a lot of structure to it. We can imagine that once we've done that like 100 times, some patterns will start to emerge. And hopefully those patterns we can easily distill among the community again. And like, here's how we are reviewing data sets or curating data sets. This is, you can do that yourself too, right?
Mhm.
Yeah.
Yeah, that's great. That's great. I understand that it's an ongoing process. It's like because it's a very complex and diverse community.
Yeah.
Yeah, but this is really good and really useful. So do you want to move to the paper?
Yeah, let's do it.
Yeah, and you also have competitions also.
We do, yeah. And we had a really nice one with ASAP Discovery or ASAP Discovery on like antivirals. That was a really cool experience. That got a lot of like, it was just a really nice like community engagement. And we did something here. Actually, if you click on this like little banner, you'll see if you scroll down to the leaderboard, you'll see that we did something which is non-obvious, which feeds into the paper, which is we have this column which is labeled like CLD, which stands for compact letter display. And what that effectively means is that we've done there are some limitations because we were in a competition setup. We couldn't precisely do what we wanted to do, but we tried to adopt the guidelines that we've written in the other paper that we'll talk about in a second. We tried to adopt them in this competition. And so an example of that is this CLD, which is we use bootstrapping to sample the performance distribution. Now, bootstrapping is flawed, but it's the best thing we could do because we were in a competition setup. We couldn't do like cross-validation because we needed to keep the test set hidden. So we did bootstrapping, and then we used statistical tests to compare different methods. And so you see, for example, that like the second and the third entry, they share the same letter. What that means is that there was not a statistically significant difference between those two methods. Whereas if we would have do the ranking in the more traditional way, we just look at the mean performance,
Yes.
we would have said that number 2 was better, right, than number 3. But what we're seeing is that it's actually not the case. Like they're similar. And so this is one of our first attempts at actually implementing those guidelines. It was a little tricky to do because we were like in a competition setup, but it's just one of the ways of showing how the product fits into the research program that we're driving.
That's cool. That's a perfect segue for the paper.
Yeah, let's do it.
There's a paper. Can you see it?
Yeah.
Okay.
Yeah, so I can speak briefly to it. So this was a great collaboration with everyone on the author list. I think a special shout out to Jeremy Ash. He's a card carrying statistician, and so his statistics expertise as well as his drug discovery expertise just was incredibly helpful in shaping this paper. Also, like a big shout out to Pat Walters. I think he wrote a blog post that sort of like seeded this idea. But really, it was really a nice collaboration with all of the co-authors. Like I think, we're all working for different companies, and so normally these collaborations can be really slow. Like no one really takes the lead. Like no one really contributes. But here, everyone was just like super excited and chimed in, and I think it made for a good paper. And so the paper is on. We propose method comparison protocols, and we'll get into what that means. But I think it's worth you taking like a small step back, and this is sort of what the introduction covers. It's when we do research. When we compare different machine learning methods or different machine learning models, we often do so to justify an investment. So in industry, when we compare different machine learning models, we have these big complex systems that like these complex platforms that drive our drug discovery programs. Swapping a model in those platforms is typically quite time consuming. And so we need to justify that investment. We need to build trust with interdisciplinary teams. We need to show that the model actually does better than what we currently have implemented. What's maybe less obvious, because sometimes we forget it, is that the same is true in research, but in a slightly different way. In research, like the scientific method, right? It's cyclic. It's not a line. It's cyclic, and so everything we put out that adds to a shared body of knowledge that others will then again build upon. And so here too, we're trying to justify an investment for all the researchers that come after us to build on the research that we're doing because we think it's a promising direction. And so both of these use cases for method comparison, it's important that we compare methods with a rigor, because otherwise we might either be pursuing research directions on the basis of shaky assumptions, or we might be investing a lot of resources in swapping out models where then in practice that model actually doesn't do better than what we currently have. So I think that's sort of the starting point. Now,
Mhm.
we already highlighted this earlier in the conversation, but in drug discovery, we're working in a different setting. Like we have smaller data sets, they are very often imbalanced, and they're typically heterogeneous. Which
Mhm.
heterogeneous, I mean to say, even large data sets are typically made of like smaller batches that are not comparable with one another, or there's at least a lot of heterogeneity among those samples. And because of that, there's a lot of stochasticity that not just comes from the model, but it also comes from the data set. And so training the same model on a slightly different split of the same data set, you might see very different performance metrics. And so again, feeding back to like these two use cases of method comparison, the important part here is that we build trust in the conclusions. And we do that by not just evaluating a model on a single split, but doing that on multiple different splits. And then now the question then becomes, okay, how do you generate those different splits? And what is what's the way to compare the numbers that you get for all those different splits between two methods? And that's what this paper is all about. But before I speak any further, like any questions at this point?
Um, Now, yes. So, not really a question, but more. So we see the figure here, the multiple comparison. You have the method and the comparison here in blue, and then you have the
Yeah.
similar ones in gray, and then you have the significantly worse in red. So I guess the CLD is basically very similar to this. So they would be these
Yeah.
ones would be the same letter.
Yeah, exactly.
Mhm.
Yeah, so it might be slightly different because of how the CLD is assigned, but the concept is the same. So the idea is that rather than having an absolute ranking where you get one, where you get like you rank them like 1, 2, 3, 4, you sort of get these equivalence classes. So like all of this performs statistically similar, and then this performs statistically significantly worse.
Mhm.
And so, yeah, it's a different way of thinking about how you compare your methods.
Yeah, and basically this is done through different runs on different splits.
Yeah. And again, there's some nuance to how we generate those splits.
Mhm.
Specifically, we'll use cross validation, a specific type of cross validation. But yeah, the main idea is we want to run multiple replicates of the same experiment to make the analogy with like wet lab science.
Great. So now we can make it slightly more complex.
Yeah, let's do it.
Yes, and get into the way. Did you want to go? Do you want to talk to the splits first, or do you want to do the basic? Not, I mean basic. I mean the fundamental
Yeah,
comparison guidelines. Which what do you want to talk about first?
yeah. So I think well, to feed back to the discussion we were having earlier, right? Like we don't just want to prescribe people what to do. We also
Mhm.
want to make it very easy. And so you'll see some tricks in this paper to emphasize that. And so the main trick you'll see is this sort of graphical um, abstract that we have
Mhm.
here. So it's formatted as a decision tree where you can sort of like follow the path through the decision tree, and you can then find a section where you can read more about that. And so the idea is there's a lot of information in this paper. People should be able to just reference it and very quickly find the section that they're interested in. And so that's where this decision tree comes from. I don't think we need to spend much more time on it, but I think that's the main reason for including that figure. You'll see there's sort of a couple boxes which we'll cover today. Like the
Mhm.
first is like the based on the data set size, like how are we going to split our data set,
Mhm.
and
Yeah.
then how, based on whether the parametric assumptions are sufficiently met, which statistical test do we use to compare methods. And then in terms of practical significance, it's all about making sure that we select metrics that reflect impact in the drug discovery context. And then finally, we talk about visualizations. Like we're generating a lot more information throughout this entire workflow. And so we also wanted to give people like easy to use visualizations to show all that information rather than having to come up with it themselves.
Yeah, yeah. This is. I really like this workflow. I think it makes a lot of sense. Especially the fact that you start with. Obviously, I like the paper. Otherwise. So,
Anyway.
But especially like the visualization, it reminds me of what my co-supervisor told me when I was with my PhD. It's like always look at your data. You might have the
Yeah.
best metrics in the world, but always look at your data. And visualization is also an art. So picking the right visualization so you get the right type of information is also very, very important.
Yeah.
Yeah.
Yeah, indeed. I think there's a lot more we can do there. And so this is the first paper. We're planning to write more. So that's currently in progress. And I agree. I think visualization, at least as a sanity check, you might not even publish it in the journal, right? But just looking at it yourself makes a lot of sense.
Yeah.
Maybe another thing that's worth noting is like you'll see that there's so you have sort of the blue path through the center of decision tree. We think that in the vast majority of cases, this is the path that people can follow in small molecule property prediction. But there are exceptional cases when your data set is, for example, really, really small or really, really large. And we
Yes.
provide guidance for those as well in the appendix. But what we really suggest is, like, I think for the sake of this podcast, like the blue path is by far the most interesting to the majority of use cases.
Yeah. It's also the size of the majority of the data sets, like 500, 200,000. That's most of the data sets.
Yeah. So it's worth, like,
I mean, I wouldn't say the most. Maybe I made a mistake because you also have lots of data sets that have very, very small size data sets that also exist.
yeah.
But let's say
Well,
most of the use
especially
cases.
so we are focused on small molecule property prediction, right? And so it's very different from like single cell or computer vision, where we have very small data sets, typically like less than 1,000 or less than 10,000 at least. And so we fall within this regime. And that's really the scope of this paper, is specifically small molecule property prediction. Yeah.
Yeah. And what do you mean by parametric assumptions?
Yeah. I think we should start with the cross-validation. I think
Okay,
that will yeah.
Okay.
So I think if you showed, yeah, that's a nice figure. So okay. So we know that with the data sets we're working with, small changes in the data set can have a drastic impact on performance. And so if you read the introduction, we sort of redefine what we mean by replicability and reproducibility, and then specifically statistical replicability. And so what we want to be able to do is, I'm paraphrasing here. I hope I don't upset my more statistically inclined colleagues, but what we're effectively trying to do is we want to be able to make sure we can reproduce results or replicate results on another split of the data that is IID. So if we in a similar data set, we want to see similar conclusions. And so the way we can estimate that using a single data set is using cross-validation. And now, why are we using cross-validation? It's because we want to do statistical testing to compare the distributions that we're sampling. And it's for those statistical tests, that's those parametric assumptions we just briefly mentioned. It's important that the samples of your performance distribution that you've sampled are independent or sufficiently independent. And so what the nice thing is with cross-validation is that your test set at least doesn't overlap.
Yes.
Now the problem is we need at least 25 samples to use it with statistical test. If we do vanilla cross-validation and we do 25 folds, the test sets may not overlap, but all of the training sets will overlap for a very large portion, like 90. I shouldn't have done this top of mind, but like 96, 92%, I think. of all training sets sort of like pairwise overlap, right? And so because of that overlap, the different samples are no longer like sufficiently independent. You can imagine that all the models trained on those training sets are very similar to one another. They learn something
Yes.
very similar.
Yeah.
And so that doesn't work. We still need 25 samples. Now, the cool thing is I never realized how big of a research area cross-validation still is. I knew like vanilla cross-validation. I didn't realize there was a whole body of research that's still ongoing to this day, trying to sort of solve that problem. And so what we landed on is we ran some experiments. We compared it to some state-of-the-art cross-validation method that is computationally not tractable, but that we could at least compare to. And we found that there was this repeated cross-validation, specifically 5x5 cross-validation, that actually is a really good approximation of the more computationally intensive and computationally intractable cross-validation technique that is sort of the state-of-the-art right now. And so what is repeated cross-validation? Well, you're seeing that in the middle of the screen here.
Yes.
So the idea is you split your data set 5 different times in 5 folds. And so the 5 folds between each of those iterations, they are independent of one another. Like they might overlap, but that overlap is less than if you were to just do 25-fold vanilla cross-validation. The benefit is that within each iteration, you only have 5 folds. And so you've drastically reduced the overlap between different training sets. And so this actually gives a really good approximation of not just the mean of your performance distribution, but also the variance. A lot better than if you were to just do like 25-fold cross-validation. So that's the main takeaway here is like you can do 5x5 vanilla cross-validation. And there are some practical considerations. For example, like how do you actually split your data set, right? Like we don't want to do random because we want to do some sort of out of distribution generalization. We don't go into detail in this specific paper, but the main arguments we make is that what we propose is compatible with more complex splitting strategies as long as you meet some very minimal conditions. And there's also the question of like, what if I need a validation set? And there too, we provide guidance. You can just split off part of your training set and use that as validation. It's a really easy thing to do. Doesn't jeopardize the method comparison protocol at all. So that's probably the safe option here. Does that make sense?
Yeah, it makes a lot of sense. It makes a lot of sense. So just to recapitulate, so you have the vanilla cross-validation A, like the figure, it's the panel is figure A.
Yeah.
I mean, it's obvious when you say it, but it's you can clearly see that most of the white rectangles are shared. So and that's what actually makes the models very similar to each other because they share most of the training set. Whereas if you look at the B, the repeated cross-validation, it's they're mutually exclusive basically. It's like
and they'll still overlap, and that's why there's ways of doing this even better. But for the sake of practicality, the 5x5 repeated cross-validation just strikes a nice balance.
Yes. Yeah. Okay, it makes a lot of sense.
All right.
So if we go back to the figure number 1, so you that's what you recommend for all data sets between 500 and 100,000 molecules.
Yeah, and so it's worth saying data set size is a continuum. Like this does mean that rather than training a single model, you need to train 25 of them, right? Well, I hope that that's possible for your final publication. I also understand if like you can't always do that. Like if you train a very large model, or I don't know, like it's worth. I just want to clarify, like we provide best practices that we expect to hold in a majority of the cases, and we think everyone, if you can, we think you should. Like training 25 models, that may sound like a nuisance, but we really think it will lead to more robust conclusions and just accelerate scientific discovery and the adoption of these models in industry. But it's worth saying that this is not a cookbook. We don't want to say that this is the only, like these are strict rules. It's like, no, there's always reasonable arguments to deviate, and we're open to that. in the lack of like a perfect solution that applies in every single scenario, we think the most important thing is to just be transparent. And we think these are important considerations, but you can deviate from them. But to answer your question, simply put, we think that for normally the sort of the data set sizes you find in small molecule drug discovery, this method will work.
25. I mean, I think it's reasonable for most experiments, because what's really important is the reason why you do all that is to get to the second box where you can get statistical significance.
Exactly. Yeah. And so this feeds back into your question of like the parametric assumptions. And so we want to use statistical. So we have 25 performance measures effectively for each of our models. So if we use MAE, you have 25 MAE scores, right? Now the question becomes, how do you compare those? And the important nuance here is that we should think of these as distributions. They're samples of the finite samples of a performance distribution. And to compare distributions, we shouldn't just compute the mean and then look at the mean. Even computing the mean and then like coming up with like error bars, like overlapping error bars doesn't necessarily imply statistical significance, or not overlapping error bars doesn't imply statistical significance. And so the important piece here is that we use the appropriate methods that have been developed, which are statistical tests, to actually compare different methods. And so that means we can no longer with certainty say whether one method is better than the other, but we can compute intuitively explained, we can think of this as like we can compute a probability that the two samples come from the same distribution, and we can reject that probability if we're below a certain significance threshold. And so that's what statistical tests do.
Yeah, that's what you're presenting figure 3 here.
Yeah, exactly. Yeah. And so we compare, like, I'm not a statistician by training, so I'm going to explain it on like a more technical level, but like we compute a t-statistics, and then we can, which is the idea that the mean of the two sample distributions that we have is the exact same. It's a measure of that. And then we can compute the probability that the t-statistic we actually observe under the assumption that the mean of those two distributions is the same. We can compute a statistic that tells us how likely that is. And if that's below a certain likelihood threshold that we set ourselves, typically 5%, then we reject the hypothesis, and we thus conclude that they are statistically significantly different distributions.
Yes. So you basically reject the null hypothesis that they have the same have the same means, and they're all basically two normal distributions with the same mean.
Yes. Yeah, indeed. Yeah.
So, okay, but that's actually, it's standard practice in biology to do things like that.
No. And so it's standard practice in a lot of different domains. And so the question is, why is it not in machine learning? And so to an extent, there are, we talk about this in the introduction as well, there are a lot of voices right now which are proclaiming, some even go as far as proclaiming like a replicability crisis, because people are like, I can't, and that's beyond machine learning. But a lot of scientists are saying like, I can't reproduce the results of other groups, or even my own group. I can't reproduce those results or replicate those results. The question is like, why machine learning? Like, machine learning used to do a lot of cross-validation, and they used to do statistical testing. So why did we let go of that? Well, the reason is that the data sets got so insanely large, specifically in computer vision and natural language processing, that on the one hand, it was just not practically feasible anymore. But on the other hand, if you have such large data sets, you can assume that your results are going to be statistically significant in most settings. You're just operating at a completely different scale. So that's why a lot of machine learning practice recently, they're not really looking into that. I think it could still be beneficial for some cases, but like there's a reason, there's a sensible reason why it's not as widespread in computer vision and NLP as it once was. That being said, we're in a different setting here. We have small data sets, they're imbalanced, and they're noisy, they're heterogeneous. So it just matters a lot more. And that's something that we should be aware of as a field.
Mm-hmm. Yes. Can you talk about why did you choose a t-test, the assumption that the values are normally distributed?
Yeah, so it's actually we're not actually recommending a t-test. So if you scroll down a little, let me see. No, it's a bit too far. Let me see. A little bit down. I think it's at the bottom of this page. Oh, here at the top left.
This
Yeah,
one?
so the top left, so guidelines too. So you can see it's what we recommended. You can think of it as an extension of the t-test. And so there's two decisions. There's a couple. I think there's three important sort of like design criteria. The first of all is like within method comparison, we were primarily looking for statistical testing with a low type 1 error rate. So low number of false positives, meaning the test will not say that two methods are significantly different while they're actually not, right? For us, it was important that all the results we observe, if we say that one method is better than the other, we actually can trust that conclusion. So that's one thing. The other thing is we're typically not doing just one comparison, but we're doing multiple comparison, right? Like I might not be only comparing method A to method B, but I might be doing all pairwise comparisons for method A, B, C, and D, which is in that case, you're doing a lot of different comparisons. And remember that we have this significance threshold. So say that like my significance is like 0.05. Like that's my false positive rate. That's what I'm okay with, 0.05. If I run that experiment like 100 times, I would expect to have 5 false positives. And if you do all those pairwise comparisons, like the number of comparisons you do, goes up quickly. And so we need to adjust for that. We need to have a correction that tells us, gives us confidence in the result of the entire suite, not just the individual comparisons.
Yeah.
And so there's various ways to compare for that. Like simply thinking, you can think of like lowering significance value, significance threshold. What we're recommending here is we're recommending a repeated measures ANOVA, and then you feed the outcome of that, specifically the squared error, I believe, but I'm not 100% sure if I remember that correctly. But like you do the repeated measures ANOVA, and that feeds into Tucky's HSD. And Tucky's HSD specifically is designed for pairwise comparisons. And so it will ensure that the family-wise error rate is below the threshold that we set.
Okay.
And so that's the important part here, is that we want to, on the one hand, we want to have a low type 1 error rate, and we want to correct for multiple comparisons. Now, there's one thing that's worth saying, which is the parametric assumptions. So in statistical testing, you have typically two families. You have non-parametric and parametric, where parametric, they make stronger assumptions about the distributions under comparison. Typically, that's why people don't like using them. It feels like non-parametric are more flexible and thus are sometimes considered the preferred option. We're actually making the case that even though non-parametric make smaller assumptions, like make less strong assumptions, they still make assumptions. You should check. And there's all sorts. There's downsides where the difference, especially if you're interested in the effect size, so not just is there a difference, but like how large is that difference? That's really hard to get from non-parametric tests, but it's quite easy to get from parametric tests. And so if you use our cross-validation guidelines, most of the parametric assumptions should be reasonably met in most applications. And so you can quite reliably use these tests. We do always like recommend checking them, and we provide guidance for that as well. But in most cases, if you do cross-validation in 5x5, you can imagine that this will work as well.
Yeah, definitely. I think you touched upon a very important aspect, the assumptions of statistical tests. I think this is this might be one of the reasons why we. I don't think we have necessary replicability crisis. I think it's too strong of a word, but I think there's sometimes a misunderstanding of the statistical tests that are applied. Yeah.
Well, I would say that like in a lot of machine learning papers, like I can't remember the last paper I've read where they actually have done statistical testing. That might be just my bubble, but like it's not common practice as much, right? That's the reason for writing this paper, is to get people to use it more. And I do think the assumptions are an important part of it. Like again, like I was really grateful for my co-author who had that statistical background, because for me, once we started sort of opening this box, there was just so much that I didn't really know where to start. And so like I would have probably just done like bootstrapping and then non-parametric tests, because those are the easiest. And so this paper is all about like, okay, that's the starting perspective. Here's why you shouldn't do that.
Yeah. It's, I was referring to more because I read a lot of biology papers, so I see a lot of p-values.
Right.
There's p-values everywhere, and sometimes it's definitely less common than it used to be. Definitely, like people got really. I mean, I can't recall exactly when the issue was, but like people have definitely there's a much better understanding of the different statistical tests and what they should be applied when. But it's sometimes the assumptions are still sometimes overlooked. Like you can just use a t-test, for example, or a Fisher's exact test. I mean, there's some strong assumptions built inside the test that don't necessarily fit the data.
Yeah.
Yeah.
So like again, like I don't have as much as a background, and so beyond what's written in this paper, I would have a very hard time arguing for one thing or another. But I'm really relying on the expertise of my co-authors here, and I think the important part is within the context of this protocol, this makes sense. And here's why, and here and an argument as to why. Now, if you are more familiar with statistics than I am, like again, like this is not meant to be a cookbook. Like, please feel free to deviate, but then explain why. Don't just like do that without any like explanation. Like just like explain why you do the testing, the comparison in a way that you do, and such that people can also easily like find that piece of information and give feedback if they think it's not the right choice, or just learn something new if they think it is the right choice, right?
Yeah, definitely. And I think you should do that. I think it should be standard practice to explain why you chose that specific statistical test.
Yeah, and there was a really nice. We did a bit of an experiment with this paper, which it's a bit of a tangent, but I think it's nice to share. Is we put this paper out as a preprint in November of 2024, and we had what we called an open feedback period. So for a period of a couple months, we had a GitHub forum set up where people could come in and share any feedback they may have. And we would read the feedback, respond to it, and we actually made some changes to our paper because of it. And I thought this was like such a nice experience. These guidelines are not just, again, I don't have all the answers. No one does. Not even our group has all the answers. And so this needs to be a community-driven thing, right? Like ideally, this is like something that's carried by the community, and it evolves as we get more and more insights. So yeah, it's just a little like tangent to say it was really nice to just have that like open review period.
Yeah, I really like it. I think it's a really good framework to start.
Yeah.
Yeah.
And probably for the majority of cases it's okay, but
Mhm.
we're not. I don't want to sit in like an ivory tower and be like, this is what everyone should do, right? It's like, no, like challenge us. Like there's a GitHub, it's still live. Like I'm pointing everyone
Mhm.
there. I can share the link with you afterwards. But
Yeah.
like, it's like, yeah, like please tell us what we got wrong because
Mhm.
that's for the better of everyone.
Yes, and at a certain point, there are things that cannot be inside a method. I mean, it has. I mean,
Yeah.
like a standard method. I mean, every paper is different. Every data set is different. Every application is different. Uh, so there are probably things that you need to change all the time. And but I like the framework. Like you start with like informed cross validation, and then you make sure it's significant, and then you compute your metrics.
Yeah.
Yeah, so relevant metrics. So my only question is, you can also compute statistics on the metrics.
Yeah.
Yeah.
What do you mean specifically?
Uh, I mean, for example, if we take like let's say a standard accuracy, like accuracy, you can have like a statistical test on accuracy.
So that's actually what we're doing here.
Mhm.
So
Okay.
we're sampling, when I say the performance sampling distribution with the cross validation, we get like one measure for each, but those measures are our metrics effectively. Yeah. So the distributions we're comparing, it's not the distribution of the data itself. It's a
Okay.
distribution of the performance of the model.
I see. I see. So you have okay. So practical significance would be more like metrics that are related to the application.
Yeah. So if you
Mm.
scroll, let me see what the figure is. If you scroll to figure 6, so
Mhm.
right. So I think this is an interesting example where what you can see is so far we've been talking about statistical significance, but the only thing that tells us is whether there's a difference between two distributions or not. It doesn't tell us what that difference is. So we might have two distributions that are very, very close to each other, but they're still statistically significantly different.
Wow.
The question we're asking in this section on practical significance is taking one extra step. It's like the difference we're seeing, the effect size, does that translate into the method having more impact in an actual application? And so that's the
Mhm.
that's the main question that we're asking here. And so we give a lot of guidance on like how you could establish that. If you scroll if you scroll up a little to figure 4, I think this is a really interesting one. And it's just an example. I don't mean to say that this should be included in every single paper. It's more about
Mhm.
like how to think about this. And so, for example, a lot of these models in practice, they're used as filters to decide which molecules to make or
Mhm.
which molecules not to make. And you can compute metrics for that. And we explain you how to do that. Now, the interesting thing is you might be training a regression model.
Yes.
And so you're not doing classification, right? Like the idea of like what to make or what not to make, it's really easy to do that in classification because it's just a classification task effectively. But you might be doing regression. Now, even if you're doing regression, we think you can still like take that extra step to show the impact that this would have in a
Mhm.
downstream application. And so you can do post hoc classification, then compute your classification metrics, and then you get a sense of the decisional impact your model would have if you use it. And what we're seeing in the experiment, for example, is that if you look at the regression metrics, we would see statistically significant differences. But if you look at the decisional impact, the picture shifts a little. And so that's an example. Another example may sound very trivial, but like MAE is a metric that a lot of machine learning scientists, reasonably so, don't like because it's very sensitive to outliers.
Yes. MEs
But
mean absolute error. Just,
yeah, exactly. Yeah, sorry. Um, it might still be worth reporting it because what's interesting about the metric is that it's in the same unit as the assay. And so if you're trying to build trust with interdisciplinary teams, a chemist can look at that and be like, oh, okay, so you're this far off, and can judge based on the unit of that of MAE whether that's a good model or not. At least, it's of course not the full picture, but at least it helps with building trust. And so we give a couple examples like these on like how to make sure that your model is not just doing better, but doing better to an extent that it actually is more, it leads to more impact in your downstream
Yes.
application of interest.
Yeah, it's significantly better because, as you said, like if you have enough data points, you can get. I mean, you get a statistical test would tell you, yeah, these two come from two different distributions, but do we care?
Exactly. Yeah, exactly. It's like, do we care? I think that's the right question. And like another example is like the idea of we can use we can use like baselines is common usage, right? We can use baselines to predict the lower bound. As we were saying before, like it would be nice if more people used scikit-learn baselines. Like that just gives us a sense of like, if we're very close to the baseline, but we are training a model that is like 1,000 times more expensive, needs all these GPUs, like, is it worth it?
Yeah.
Right? Are we really like, is it worth it? Do we care? And similarly, if you know the error rate in your assay, the noise level, if you do better than noise level, if your MAE is lower than your noise level, you should question your results. Like, am I overfitting to the error in my data set? Like, is that still impactful? Like, probably not, right? And so there's things like these that we suggest on just, it's more of the drug discovery perspective. Like,
Yes.
Given how this model is going to be used, you should be looking at these things.
Yes, I think it's especially relevant for drug discovery. Especially relevant because you have at the same time you have like huge models and you have like very small models, and sometimes they're competitive with each other. So yeah,
and it matters, right? Because you're probably running these models like on a daily basis, if not more. And so you don't want to have huge compute costs if you can do like 98% of the way with very cheap models, right? So, and again, that's like the industrial perspective that sometimes I think is lacking. Or you might want to do these like ultra large virtual screens and like screen like billions of molecules. Like if you can do that with a random forest and it runs on a single CPU somewhere, that's the preferred choice. Yeah.
Yeah, you don't necessarily want to pay billions to do that when you can do it like
exactly.
Yeah. And I think it comes from the fact that the data sets are actually very different, especially if we're talking about small molecules. They're small. They're very short molecules. And the same is true also for proteins. You can have, if we're to compare, for example, a protein of a small molecule to a book, like the book has inner coherence. The coherence is different. Like in context of a protein or a molecule, it could change, for example, 1 or 2 words, and that would make completely different protein and a completely different molecule. That would never happen in NLP.
Exactly. Yeah. You have exactly. But like it feeds back to like the data we're working with is no longer intuitive. Like people spend their entire academic careers trying to understand this data. Like it's also the reason why we can't do data augmentation. Not really in like chemistry, right? We can't just like swap out the scaffold or swap out like an atom and we assume that performance will roughly be equal. It's like, no, it doesn't work like that. Like small changes can drastically disrupt the profile.
Yeah, you can just translate the thing. Like you take
exactly.
the cycle, translate it here.
Yeah, just rotate it. Yeah, it's like, no, that actually matters. Yeah. But it is again, like we're laughing about it now, but like I had that thought, right, coming in. I'm like, okay, well, you're telling me that you're telling me that like a scaffold is the core molecular structure. And so what if we just keep that the same and we switch everything around it? Then it should be roughly similar, right? We might do it wrong in some cases, but it's roughly similar. It's like, no, like
No,
No,
you can't do
it
that.
it depends, right? Like it
Yeah,
depends. Yeah.
exactly. It depends. That's the keyword. It depends.
Oh, I was so my first year as an intern, I was like fed up whenever people would tell me that. I'm like, I yearn from some straightforward answers. Please give
Yeah,
me straightforward answers. Anyway, that's where this paper comes in. Three
yeah,
3 years
me too.
later.
Me too. Exactly same experience. It's like, what do you mean? It depends.
Yeah.
No, actually, that's the right answer.
Yeah.
And proteins are so proteins molecule. It's so counterintuitive. Um,
Yeah.
they don't have symmetry. They behave very strangely. They have receptors that also don't have symmetry. It's and symmetry is such an important thing in how we process information as human beings. It's really a fundamental part of our intuition. It's like if this works here, then it works there. That's basically how we. It's like let's take like a simple example. That's how we can transform a vase into a cup. We just like pour water and drink out of it. It's like that's a form of symmetry.
Yeah. Yeah.
But that thing doesn't exist like when you go into these realms. There's
Yeah, or at least it's
it's
not
different.
intuitive.
Yeah.
Yeah. Like I do think it exists
Mhm.
to an extent. It's just it's hard if yeah, it's not
Yeah.
like computer vision and NLP,
Mhm.
everyone in the world is trained to do that from the moment that they're born. Like
Mhm.
that's what we're as humans, that's what we're really good at. And
Yeah.
so those tasks are easy to or intuitive and
Mhm.
thus easier to evaluate. Like I can look at the output of a model and understand what it does well and what it doesn't do well because I'm an expert.
Yes.
That's no longer the case with chemistry or biology or physics
Mhm.
or yeah.
Definitely. Definitely. I think you're right. I mean, it does exist, but it's a fundamentally different type of symmetry than the symmetry I mean, we're like let's say sensory accustomed to through our senses. It's
Exactly.
yeah,
Yeah.
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 It's and that's what makes it cool and super interesting.
Yeah, and I've worked with great chemists, and they will have that intuition, but it's built up over 40 years of experience, right? Or 20 years of experience, but like it's built over years of experience. And they will look at my predictions for a molecule and they'll be like, well, that's not never going to be soluble, or like that doesn't make sense. I'm like, oh, okay, cool. But then I asked them, like, can you explain it to me? And they're like, I'm not sure. It's like it's sort of like this fuzzy intuition. Like sometimes they
Yeah.
can pinpoint to certain things, but like a
Mhm.
lot of it is from like earlier experiments they've run that they were part of that they sort of vaguely remember the results of. Like they build up that intuition,
Mhm.
but again, that takes years.
Mhm.
So
Yeah.
we yeah we need to, I think, as a community come together and like just make these guidelines easy to use and readily available such that people don't um have to reinvent the wheel all the time.
Yes. Yes. And as you said, it takes years and years usually to develop that intuition. It takes yeah, it takes like seeing like thousands and thousands of molecules in thousands and thousands of settings, and then you somehow your brain get accustomed to predicting this kind of stuff. Um, whereas for language and imaging, we have basically we have we've been doing that for so long, like millions of years. Actually, it's like our brains have been designed to process images for millions of years. So that's why we're also so
Exactly.
good at at evaluating the quality of the data set.
Yeah, exactly.
Hmm. Yeah.
Maybe as a like the
Hmm.
last section of this paper.
Hmm.
So as I was saying, like we produce additional
Hmm.
information um, uh, throughout this protocol. So if you scroll down to like figure 7,
Hmm.
yeah, that one, the one above.
That's 8. Yeah.
Yeah. So we propose. Oh, sorry. I actually meant. I'm in a different one. I meant. No, that's the one. Figure 7. Yeah. So um, there's additional information here. So we have a couple of different things we want to show. We want to show statistical significance, right? Like, is there a difference that is statistically significant? But then we also want to show the effect size. And so now we have two things we need to show. And the question is, how do you do that? And there's a variety of ways to do it. I'm not as opinionated about this section as I am about the earlier section. This is more of like, this is a starting point, take it or leave it. But feel free to come up with other visualizations that you think are better. This is one of the examples. It's the one that we saw in the graphical abstract all the way at the top of the paper. And so what we do here is we take the method that we've proposed, that the method that we're specifically interested in comparing. We color that in blue, and everything that is similar to it, we color in gray. And the nice thing about this visualization is if the error bars overlap, the error bars have been scaled in such a way that if they overlap, it tells you that there is not a statistically significant difference. And so we can, and then using color, we can indicate which of the methods are similar, which of the methods aren't. Now, one of the things we're not seeing here is we're not seeing the effect size. So we're seeing the difference in the mean performance, but we're not seeing the effect size. And so if we go to another the plot, the one below, the heat map, so you can see here we've sort of turned it around. And what we're doing here is because we think effect size is really important in these comparisons, we're showing that first. We're showing that inside of the box. So you can see, I don't know, for the third row and the first column, you can see that those boxes are turned blue. That indicates a certain effect size. We are also indicating statistical significance, but the way we're doing it here is with little stars. So we're not giving the precise statistical significance, we're just giving those little stars. It's two ways of visualizing the information that we have, and you put the emphasis on different parts. There's pros and cons to each. I'm not as opinionated here, but I do think it's just important that we know what pieces of information we can visualize and to share some ideas on how to do so.
Yeah. Second one looks like a standard figure you'd see in a biology journal.
Yeah.
Heat map with stars.
Yeah. And so there's different ways of doing it. I know we didn't recommend that, but I've also seen like bar plots, and then they use the stars with the annotations. I think that doesn't work as nicely, or like box plots, and you do the same. But anyways, again, I'm not as opinionated about this part, but I do think it's important to realize what you should be showing and to give an example. Now, there are cases where I think one thing that's worth saying is we're explicitly not recommending a leaderboard, right? And like a leaderboard or like a table, the table with like a row in bold to show what's best, like that's currently the standard, and we're actively arguing against that. Now, if there are cases in which you really, really, really want to show a leaderboard, and that could be, for example, because the reviewer asks you to, your reviewer number 2 might ask you to.
It's always number 2.
Sorry? Yeah, it's always number 2. Yeah. Exactly. So like that could happen. Then we do provide guidance using that compact letter display that we showed on the platform early on. Like for us, that was the only way to do it because we couldn't do cross-validation in the competition setup. So we sort of landed on that. But anyways, it's just to say like there's alternatives that you can use here.
Yeah, or you can still do the table and then put it in supplementary.
Yeah, exactly. Yeah. Yeah.
That's cool.
So yeah, and then the last thing is just to conclude. There's actually 2 more things now that I think of it. So we have section 4. It's very brief section, but the important piece of section 4 is to say that this is just another visualization.
Yeah.
Shows the same information in a slightly different way.
Yeah. I think like the Space Invader.
Yeah. I had not realized, but yeah, I see that now. Yeah, like there's 2 more sections to the paper. Section 4 is we have open source code. Again, we don't just want to like prescribe, but we want to make it easy. And so we have open source code using open source libraries to implement all of these things in Python. This is also the link where you'll find that forum that I discussed, where people can give feedback on the guidelines themselves.
Mhm.
So that's kind of interesting. And then section 5 is really simple. It just summarizes the paper. And specifically, it lists those guidelines again. So yeah, that's the main gist of the paper. And so there's more in the works. Like this is method comparison is one part. We think there's more to do, like how to split your data set, how to curate your
Mhm.
data set. Like all of these are questions that have few straightforward answers.
Yes.
And so we would like to do more of these papers in the future.
Great. I think I would definitely read them.
I love to hear it. Yeah.
Yeah, I think this is a very pretty cool contribution to the community.
Thanks. Yeah. We're also hopeful. I mentioned like we need to create incentives, right? And we just joked about reviewer too. Like I think that's an important piece that's now missing. Like
Mhm.
reviewers and journals and conferences, they sometimes ask you to produce things because that's been the standard for a long time, which is
Mhm.
actually not that scientifically useful, or it's not as useful as it could be, right? Like they'll ask you to include a leaderboard, even though that leaderboard probably isn't the best way of visualizing a method comparison. And so what we're really hoping by publishing this paper as well, and I was really happy that we received the editor's choice from JCIM, is that it also like it creates a paper to fall back on for reviewers to also review journals and to start changing those standards, right? I want to see papers that, of course, I'm biased, but I would love to see papers that do stuff like this more and more. And so I'm hoping that having the paper out there also it's a reference for for the community at large, that we can like if you submit a paper, you get an annoying question for reviewer two, that this is something you can reference and be like, well,
Mhm.
actually, there's this whole industry consortium that said otherwise, and here's a published article in GCAM
Yeah.
that documents that.
Mhm.
Yeah,
Yes. And that's also the standard way. It's also a very important way science evolves. It's like, how do we
exactly.
communicate science to each other? It's
Yeah,
yeah, yeah.
yeah.
It's a constant evolution. It's a slow evolution, but it's a constant evolution. If you look at papers from 30 years ago, the data is represented differently.
Yeah, exactly. Yeah,
Mhm.
and I think the journals are an enormous driving force. They have an enormous
Mhm.
power there, and the conferences too, right?
Yeah,
Um, so yeah,
definitely. So we can finally move out of the leaderboard with the bold
I
model in bold.
I hope so. I'm done. I'm done with those papers, especially if there's like no variance on it, like
Yeah.
not even an estimate, not even using different model seeds or something. I'm just like, oh
Mhm.
yeah.
Yeah. Yeah. Yeah. Yeah. I think that's why I like this paper so much. It's like finally. there's a there's a good standard that we can start working with. Do you want to talk a
Thanks.
little bit about biology and living
Um,
cells and just to have
I
time for that?
No, I actually have to hop off. I
Okay.
have some other meetings to end my day.
Mhm.
Do you want
Yeah.
to do like an outro of some sort, or
Yeah, sure. Do you have anything that you'd like to talk about, reference where people can find you,
uh,
websites like Polaris Hub?
I'm not very active. They could probably,
Mhm.
yeah, huh? I probably should have prepared that, huh? No, I'm not. I'm not very big on social media. I don't like. I always feel like an
Oh,
imposter when I try to make a name for
that
myself.
doesn't
But
have to be social media. It could be just like your paper, could be Polaris, could be anything you want.
well,
Um,
I would love like I think any
mhm.
feedback that people have. I think
Mm.
that's the main ask here, right? Like
Yeah.
we've done our very best to present something here that we think is reasonable.
Mhm.
Despite our best efforts, you might disagree
Yes.
with us, and we welcome that disagreement. If you think there's something that would like drastically improve this paper, the best way to leave your feedback is to go to github.com, like the link you've selected right now, actually. If you go to that link, you'll see that there is a, what's it called? Like, I think they call it a wiki? No, discussions. So we've enabled discussions there. And what you'll see in those discussions, that there's already several discussions created where people challenge some of the things you've said. And you see in some cases that has led us to revise the paper or change it. Like, please share any feedback you might have there. Like, I'm sure there's things we can do better. Other than that, like the Polaris platform at polarishub.io, we're still working on this. We want to really now take the next step. Now that the paper is out, we really want to take the next step in implementing the guidelines we've talked about here directly into the platform. There's a really nice blog post we put out where we describe the idea of an evaluation dashboard. Yes. Where you can do these statistical tests and you no longer present it with a leaderboard, but you can actually do these statistical tests interactively through the dashboard. That's a big feature. I'm not sure how much time it will take us to get there, but I'm really excited about that. Like, I think we can now really start changing how people do evaluation. And if anyone else wants to get in touch with me for anything else, not very active on social media, but LinkedIn is probably the best way to do so.
Or like, not enough people come here actually into social media. That's
yeah. I probably should be a
You're
little
not the only
better.
one.
Yeah, like I think when you do your master's degree or your academic academic degree, you're told that like it's what is it, like content over form. Like it depends on the content is what's really important, not really how you package it. And I think that's such a naive take.
It is.
I really don't think science works like that. Like, I wish it did. I wish we all judged each other on the quality of our writing, right? But no, like there's so much out there. You need to figure out a way to stand out and to make things easily digestible. And so I wish I was a little better at the social media game. I think I would get more traction, but it's fine. I have Pat Walters on this paper, and he's great at it. So.
He's good. You're absolutely. It's actually a skill. Like,
and it's not. I don't mean to say that people that do it well that they're not good scientists. Like, I agree with you. Like, it's a skill, right? Like, it's like that's part of your job as a scientist. You can't just write a paper, publish it, and take your hands at it. No, like part of your job is to teach people and to share that knowledge and to be open to feedback and to improve and to work together. And like, that's all science as well.
Yes, that's very true. Very true. So, man, but the thing is, it's when you do science, it's usually on a different time frame. It's like it takes time to do the research, time to write the paper, and social media usually is on a very, very fast time frame. So that's
great.
But I agree with you.
All right. And we're saying that while recording a podcast. So this is us trying to be more social.
We're doing our part.
Exactly.
Definitely doing it.
Spreading the gospel.
Yeah. So,
anyways, thank you so much.
Thank you, Cas. It was great. Very insightful. I really appreciate it. I think it's an awesome podcast.